Hi, my name is Neil Gandhi and I'll be presenting my final project on cell structure. This video will include 3 parts. 1. My slides, 2. A model that I made, and 3. On my thoughts and understanding of cell structure.
What is a cell? Cells are building blocks of life. Every living thing, plant, animal, and human, is made of a cell. Some organisms are just made of one cell, but humans have trillions. There are 2 main types of cells, prokaryotic and eukaryotic. Prokaryotic cells like bacteria are small and simple, they don't have a nucleus. But eukaryotic cells, which include animal and plant cells, are more complex. They have a nucleus and many other organelles. In this project, I focused on animal cells, which are cells found in human bodies. Slide 2 talks about what cells even are. Cells are all living things are made up of cells. Cells are the basic unit of life. They all do important jobs to keep the organisms alive. The 2 different types of cells are plants and animals. The first one is plant. They have a cell wall that gives them shape and support. They also have chloroplast, which helps them keep are attached to the rough ER. Liposomes are small sats that break down waste. And why this topic is important in biology is because it reveals how cell functions at a molecular level, which is crucial for identifying what goes wrong in diseases like cancer and infections. Structural insight are key to drug development, as many new target-specific organelles or structural proteins. Innovations in imaging and structural biology are driven by a new era in precision medicine. Cells, where treatments can be tailored based on cellular
All living things are made up
of cells. Cells are basic units of life, and they do all important jobs that keep organisms alive. There are 2 different types of cells, plant and animal. Plant cells, these have a cell wall that gives them shape and support. They also have chloroplast, which helps them make food using sunlight, which is also called photosynthesis, and a large vacuole that stores water and nutrients. The second one is called animal cells. These don't have cell walls or chloroplast. Instead, they have a small parts called liposomes that helps them break down waste inside the cell. Some tiny organisms like parasites have special features to help them survive. One example is Luisi, a parasite with a special surface that helps them hide from the immune system of the animal it affects. Why cells matter. Learning about how cells are built and how they work is super important for science and health. Helps scientists understand how our bodies function, how diseases like those caused by parasites happen, and how to treat them. This knowledge also helps with genetic engineering, changing the genes of plants, animals, or even humans to improve health or create better crops. By studying cells, scientists can see how life has changed over time, which is also called evolution, and find better ways to keep people healthy. Animal cells and their structure. Animal cells don't have cell walls like plant cells do. Instead, they're surrounded by flexible cell membrane. Cell membrane allows animal cells to have many different shapes, unlike plant cells, which are more rigid. The flexibility membrane also helps them easier for animal cells to move, change shape, and communicate with other cells. The cell's membrane acts like a gatekeep that controls what goes in and out of the cells, like nutrients, water, and waste. This helps the cell stay healthy and respond to change in its environment. Trypanosoma. Cells don't work alone. They send and receive chemical signals to help stay connected and coordinate their actions. These signals help cells know when to grow, divide, move, or respond to threats. This process is important in single-celled organisms, which use signals to find food, avoid danger, and is also important in multicellular organisms, where the cell must work together in complex systems like the brain, immune system, or muscles. Studying how cells communicate helps scientists understand disease caused by broken signals, such as autoimmune disorders, cancers, and certain infections. Cell surface in trypanosoma. Trypanosoma is a blood parasite known for its special surface sugars. These sugars cover the outside of the parasite and act like a shield, helping it avoid detection by the host immune system. Because of its clever adaptation, this parasite can survive longer and grow inside the host's blood without being easily destroyed. This makes it harder to treat and control infections caused by this parasite and others. Cell wall presence in cells. Yes, we've already went over this, but yes, plant cells have a cell wall, a rigid outer layer that provides structural support and shape to the cell. This cell wall is primarily composed of cellulose, a complex carbohydrate. It helps maintain the cell shape, provides protection, and allows cells to develop tertiary pressure. Animal cells. Animal cells do not have cell walls. They are surrounded by a cell membrane instead of many animals' tissue having an extracellular matrix that provides support and communication between cells. The second question is it ethical to use animal cells in model research or testing treatments? The pros of this are genetic animal cells share many features with human cells. Parasites work, so testing on find ways to stop diseases help predict how treatment may lead to new treatment in real bodies. The cons is using animal cells accidentally or using animal means living creatures which can suffer during its harm. Ecosystem ethics are researchers follow issues. Ethical guidelines to reduce harm, like follow strict safety rules and be done responsibly with careful cultures to weigh when possible and refining methods to minimize suffering. The third question is, how do we balance innovation and potential misuse in cellular research? Innovation benefits. New discoveries about cells' function, gene editing, and cellular signaling can improve medicine, biotechnology, and disease control. The risks are advances in cell manipulation could be misused to create harmful biological agents or cause unattainable damage to living cells. The balance is strong ethical oversight, regulations, and public discussion help ensure cell research is used safely and responsibly. The future direction in cell research. The future cell research direction includes personalization medicine, advanced tissue engineering, and the development of artificial organs. Stem cell research is also driving process in areas like gene editing, regenerative medicine, and the use of stem cell for drug testing. Personalization medicine. Doctors can customize treatment based on how a person's cell responds, making medicine more effective and safer. Tissue engineering. Scientists are learning how to grow tissue and organs in the lab to replace damaged body parts. Artificial organs. Researchers aim to create fully working artificial organs to help patients in need of transplants. Stem cell research. Stem cell research can help turn into many types of cells, helping repair injuries and test new medicine. Gene editing tools like CRISPR allow scientists to fix genetic problems in cells.